Hello everyone. Today we're going to dive deep into the fascinating world of DNA sequencing. Specifically, we'll explore first-generation sequencing. We'll discuss its working principle, see how it works, and learn about the situations when it's the best choice. Finally, we'll talk about its applications and advantages. Let's get started. So, what is first-generation sequencing? First-generation sequencing, also known as Sanger sequencing, is a DNA sequencing technique based on the chain-termination method. It was invented by British biologist Frederick Sanger in 1977, hence the name Sanger sequencing. This method was the first widely adopted technique for sequencing DNA and played a critical role in the early days of genetic research, and Frederick Sanger was awarded the Nobel Prize in Chemistry in 1980 for his work. Sanger sequencing laid the foundation for modern genetics research and is widely used for research purposes like targeting smaller genomic regions in larger number of samples, sequencing of variable regions, validating results from next-generation sequencing, or NGS studies, verifying plasmid sequences, inserts, mutations, HLA typing, genotyping of microsatellite markers, and identifying single disease-causing genetic variants. Sanger sequencing is based on synthesis of DNA strands that are complementary to a template DNA strand. The sequencing reaction uses normal deoxynucleoside triphosphates, or DNTPs, and modified dideoxynucleoside triphosphates, or DDNTPs, for strand elongation. The DDNTPs are chemically altered with a fluorescent label and with a chemical group that inhibits phosphodiester bond formation. DNA polymerase synthesizes the new DNA strand, occasionally adding a DDNTP instead of a DNTP, which terminates DNA synthesis. This creates DNA fragments of various lengths, each ending with a different specific DDNTP. The resulting DNA fragments are subjected to capillary electrophoresis, where the fragments flow through a gel-like matrix at different speeds according to their size. Each of the four modified DDNTPs carries a distinct fluorescent label. The emitted fluorescent signal from each excited fluorescent dye determines the identity of the nucleotide in the original DNA template. Next, let's delve into the detailed steps of how first-generation sequencing is performed for DNA sequencing. Here's a step-by-step description of the process. First is sample preparation. The first step is to obtain a sample of DNA that will be sequenced. This could be from a variety of sources, such as blood, saliva, or tissue biopsy. The DNA must then be purified and concentrated so that it can be used for sequencing. Second is fragmentation. Initially, the DNA sample is fragmented into smaller pieces ranging from around 100 to 1,000 base pairs in length. This fragmentation is crucial for making the sequencing process more manageable. The process begins with a mixture of numerous DNA template strands, primers, DNTPs, and DDNTPs. Next is primer annealing. Primers are short, synthetic DNA sequences specifically designed to be complementary to the target DNA region intended for sequencing. These primers contain standard deoxyribonucleotides, or DNTPs, that enable stable binding to the target DNA. The primers are mixed with the fragmented DNA, and they randomly anneal or bind to the target DNA at their 3' ends, which are complementary to the 5' ends of the target DNA strands. The mixture is then heated to facilitate the annealing process, allowing the primers to effectively bind to the respective target sites on the DNA templates. Fourth is synthesis. Following primer annealing, the next step is primer extension, where nucleotides are added to the primers in order to synthesize the complementary DNA strand. A DNA polymerase enzyme facilitates this process by attaching individual deoxyribonucleotides, or DNTPs, and chain-terminating DDNTPs to the 3' end of the primer, creating a growing DNA strand. The DNTPs and DDNTPs randomly bind to their corresponding complementary nucleotides on the template. Since there are countless template strands, the final products include numerous possibilities with different fragment lengths. This technique is referred to as sequencing by synthesis. Fifth is termination. During the synthesis phase, a special class of nucleotides known as chain-terminating DDNTPs are introduced into the reaction mixture. These chain-terminating nucleotides lack a 3' OH group required for the formation of a phosphodiester bond between two nucleotides, causing DNA polymerase to cease extension of DNA when a modified DDNTP is incorporated. The DNA sample is divided into 4 separate sequencing reactions containing all 4 of the standard deoxynucleotides: DATP, DGTP, DCTP, and DTTP. The DNA polymerase and one of the 4 dideoxynucleotides, DDATP, DDGTP, DDCTP, or DDTTP, in each reaction. Next is gel electrophoresis. The products of the reaction are separated by size using denaturing polyacrylamide urea gel electrophoresis. For each of the 4 reactions is run in one of 4 individual lanes. Lanes A, T, G, C, and the DNA fragments are separated based on their size, with the smallest fragments migrating the furthest length through the gel. Next is imaging. After gel electrophoresis, the DNA fragments are visualized using a process called autoradiography, which involves exposing the gel to X-ray film or detecting the bands under UV light. The dark bands correspond to DNA fragments of different lengths. Next is sequence determination. The final step is to determine the sequence of the DNA fragments. This is done by reading the order of the nucleotides in each fragment and comparing it to a known sequence of the primer. A dark band in a lane indicates a DNA fragment that is the result of chain termination after incorporation of a dideoxynucleotide, DDATP, DDGTP, DDCTP, or DDTTP. The relative positions of the different bands among the 4 lanes from bottom to top are then used to read the DNA sequence. By examining the bands in each of the 4 lanes, the DNA sequence can be directly deduced from the gel image or X-ray film. Sanger sequencing can be used to identify and confirm COVID-19 subvariant types by sequencing specific regions of the SARS-CoV-2 genome, particularly the spike protein, where many functionally relevant mutations are found. Here's a detailed explanation of how Sanger sequencing can be employed for this purpose. Sample collection. A nasopharyngeal or oropharyngeal swab is collected from a patient suspected of having COVID-19. RNA extraction. The viral RNA is extracted from the collected sample using an RNA extraction kit or other appropriate methods. Reverse transcription. Since SARS-CoV-2 is an RNA virus, the extracted viral RNA needs to be converted into complementary DNA, or cDNA, to be compatible with the Sanger sequencing process. This conversion is done using a reverse transcriptase enzyme in a process called reverse transcription. PCR amplification. Specific regions of the SARS-CoV-2 genome, such as the spike protein gene, are targeted and amplified using polymerase chain reaction, or PCR. This process involves designing specific primers that bind to the target region, allowing for amplification and generation of multiple copies of that specific DNA fragment. Sanger sequencing. The PCR amplified DNA fragments are then sequenced using the Sanger sequencing method. The resulting fragments of different lengths are separated using capillary electrophoresis, and the DNA sequence is determined by analyzing the fluorescence emitted by the DDNTPs at the end of each fragment. Sequence analysis. The obtained DNA sequences are compared to a reference SARS-CoV-2 genome to identify any mutations present in the target region. These mutations can be indicative of specific subvariants or lineages of the virus. Specialized bioinformatic tools and databases, such as GISAID or NextClade, can be used to identify the sequences into specific subvariants. It is worth noting that next-generation sequencing, or NGS, has gained popularity as the preferred method for large-scale genomic studies due to its high throughput, efficiency, and versatility in its sequencing entire genomes or transcriptomes. Since NGS typically produces shorter read lengths, it is more cost effective for large scale products and applicable to a broader range of research areas. However, Sanger sequencing still holds advantages in certain situations. As a mature and reliable technique, it is known for its high accuracy and relatively long read lengths, typically between 500 and 1000 base pairs. Sanger sequencing is often the method of choice for specific applications such as validating particular mutations, targeted sequencing of a select few genes or regions, and instances where the high sequencing accuracy is crucial, like in some diagnostic tests. In summary, although NGS has become the go-to method for many applications, Sanger sequencing remains valuable for confirming specific mutations or validating NGS results when needed. There are several products on the market based on Sanger sequencing technology. Here are some major Sanger sequencing products and platforms. ABI 3500/3500XL Genetic Analyzer. The ABI 3500/3500XL Genetic Analyzer is a versatile Sanger sequencing platform capable of detecting various genomes from different organisms, including humans, animals, plants, bacteria, and viruses. It is primarily used for targeted gene sequencing, mitochondrial DNA analysis, microbial identification, viral genotyping, and human leukocyte antigen typing. Seq Studio Genetic Analyzer. This user-friendly instrument excels in targeted gene sequencing, mitochondrial DNA analysis, microbial identification, and viral genotyping. Beckman-Coulter CEQ8000. The Beckman-Coulter CEQ8000, a capillary electrophoresis-based Sanger sequencer, is also capable of detecting and analyzing various genomes from diverse organisms. This automated and rapid system is particularly useful for targeted gene sequencing, mitochondrial DNA analysis, microbial identification, and viral genotyping. In conclusion, Sanger sequencing is a reliable and accurate method that has facilitated a wealth of discoveries in the fields of genetics and molecular biology, playing a crucial role in projects such as the Human Genome Project. Thank you for watching.